My name is Megan Dewick. I'm a scientific solutions consultant with Benchling, and today I'm going to be talking to you about how Benchling can streamline and amplify your complete CAR-T R&D pipeline. First, I want to talk to you about how Benchling can help a lead discovery scientist generate promising leads faster. Designing and assembling new plasmids can be tedious and time-consuming. To help alleviate this bottleneck, Benchling offers a bulk assembly tool for generating groups of plasmids in a combinatorial fashion. When running high throughput screens, it can be difficult to track which results correlate with which sequences. Benchling allows a scientist to automatically link diverse result types back to their corresponding genetic parts. Additionally, using Benchling native analytics, a scientist can build custom dashboards across diverse data sets to quickly identify the most promising leads. Today, my CAR-T demonstration will begin with the creation and screening of CAR-containing plasmids. To get things started, I'm here in the Benchling registry to get some more information about a very specific CAR-containing plasmid. Now, the Benchling registry is a centralized database for all of your samples, your sequences, your plasmids, your viruses, and all of their associated metadata. Now, let's say that I want to find a very specific plasmid that contains a particular CAR gene. To find this plasmid, first I can set a filter so that I'm just looking at my registered plasmids. And now I can set a filter for my specific CAR gene. This has brought me to the exact plasmid that I was looking for. Now I want to get some information about the exact CAR gene that's in this plasmid. Here we are in the registration page for my plasmid of interest. These blue chips that you see in some of the metadata fields are links to other registered items. In this case, these other registered items are genetic parts within this plasmid. Let's click into this CD19 CAR1 sequence to get some more details about the components of this exact CAR gene. Now, all of these parts metadata fields in both the plasmid and now in this CAR gene can be autofilled by Benchling. This will both save you time and also ensure that you have sequence level accuracy. To kick off my CAR plasmid screening activity, I'd first like to bulk assemble some additional CAR-containing plasmids in Benchling. Here I have set up an automatic bulk assembly where I will be making every combination of 2 plasmid backbones, 3 promoters, and 2 CAR genes. Once I have told Benchling how to attach all of my various ends, I simply go to Finalize and I hit Assemble. Here are the 12 new plasmids that we just bulk assembled. If I click into one of these plasmids, I can now easily autofill the metadata fields for promoter and CAR sequence. Having this seamless integration of molecular biology tools with other Benchling applications removes the need to jump back and forth between programs, and now I'm ready to run some experiments on these new plasmids and also capture results against them in my Benchling lab notebook. Let's open a partially filled out lab notebook entry where I already have a high throughput CAR screen in progress. In this particular activity, I have electroporated a reporter cell line with an assortment of CAR-containing plasmids, and now I am looking at the specificity and activation potency of the various CAR-expressing reporter cells. In this first results table, I have captured activation data across varying target to effector cell ratios. In this second result table, I have captured data on reporter cell activation levels when exposed to both target and off-target cells. Now let's jump into Benchling Insights to help us make some sense of all of these data. In this top row, I have plotted my specificity assay, where CAR activation is on the y-axis, the various CAR plasmids are on the x-axis, and the various colors are representing different on and off-target cell types. These data are also being shown in table form in the top left panel. In the second row, I'm looking at all of my various dose-response curves. Now, as they stand, there is just way too much information in both of these graphs for me to make sense of anything. So let's set up a specificity filter to just look at CARs where the activation level between on-target and off-targets is over a certain threshold. You can see now I have narrowed in on the 4 most specific CARs. Now let's do some similar for our activation. Let's set up an activation filter to just look at dose responses that exceed a specific magnitude. And now you can see, after doing this, you can see that we've surfaced that both for specificity and for activation, the most promising leads happen to be CAR1, CAR9, CAR14, and CAR15. And so this is letting me know that these are the leads that are going to progress to the next stage of screening. After running an additional flow cytometry experiment with these 4 CAR leads, I maxi-prepped the corresponding plasmids and moved these plasmid maxi-preps into the inventory. Both of these things were done directly through my lab notebook entry. Now let's make a request to a downstream cell engineering team to have them make viral stocks with my 4 lead CAR sequences. So here I have selected all 4 of these plasmid maxi preps that I have just made, and now I'm making a request to my colleague, Danny, and I'm asking her to do viral production from a plasmid, and I'm going to give her a due date. Let's say I want it in 3 weeks, and I could also leave any comments here. So now Danny will receive this request and know exactly what to do. Next, I want to talk about how Benchling can help a cell engineer optimize and streamline their processes through built-in reagent tracking and in-platform analytics. Benchling provides complete sample context so that all relevant and necessary information is easily accessible to the cell engineer. Second, Benchling allows a scientist to track critical reagents and samples in real time. This ensures that location and quantity information is always up to date and easy to find. Lastly, with our in-platform analytics, the cell engineer can monitor and examine success metrics across multiple production runs. So let's go ahead and take a look at our previous request and see what that's going to look like to our downstream cell engineering team. Here is my dashboard of any requests that I have made or any requests that people have assigned to me. Now let's open the request that we just made. This request contains all of the information that my cell engineering colleague Danny needs to complete this task. In this bottom table, it lists both the exact plasmid prep that I want her to use, as well as the exact box where it is located, and also the exact tube where it is located. Let's go ahead and click into this box and find the tubes that Danny needs to use for this viral production request. In this view, Danny can easily find the tube IDs that she has been assigned. From here, she can also check out these tubes to let her colleagues know that she is going to be using them. Once Danny has completed this request to make CAR-T viral stocks, she could also then submit a new request to have her viral stocks transduced into donor cells. So let's actually jump into a fulfilled request where exactly that has happened. If I jump into the Fulfillment tab for this request, you can see that there is a direct link to the notebook entry in which this request was fulfilled. For this particular transduction, I opted to test out 3 different expansion medias. In this first table, I registered the various CAR T cells that I made. In the second table, I captured results pertaining to both CAR T growth and yield. So let's go ahead and take a look at these results in Benchling Insights. In this dashboard, I have opted to look at my cell growth data in a variety of different ways. In the top right panel, I'm looking at what percentage of my total cells were CAR T cells at varying time points. And in the bottom 2 panels, I'm looking at the growth dynamics both for total cells on the bottom left and CAR T cells on the bottom right. Let's narrow in on just one particular viral lot and compare our different expansion medias. So for this particular viral lot, it appears that media B might have given me the best overall CAR T yield. Now let's talk about how Benchling will streamline a translational scientist's path to making impactful CAR T insights by integrating results from across the entire organization's R&D, the organization's complete R&D pipeline. Now, within Benchling, a translational scientist has easy access to all of the upstream sequence based information. This allows the scientist to confirm sample identity and track sample lineage. To regiment experiment setup and results capture across an organization, Benchling gives users the options to start notebook entries from standardized templates. And finally, using Benchling Insights allows a scientist to create custom dashboards that will aggregate data from early high throughput in vitro screens all the way through to translational in vivo studies. So let's jump into an entry where we have tested engineered donor CAR T cells produced in our previous activity. Now, as with all of the other notebook entries that I've shown you in this demonstration, this particular entry could have been created from a template where all of the relevant steps and data tables would have already been established and ready to fill out. In this entry, we have captured dose response and specificity data for various lots of engineered CAR T cells. Let's go ahead and take a look at these data with our in-platform analytics. So as with the other dashboards, I'm looking at my data in a variety of different ways. So on the top right, I'm looking at activation as measured by IL-2. I've also looked at specificity with regard to IL-2, and I've also looked at both activation and specificity as measured by interferon gamma. As with the other dashboards that I have shown in this demonstration, I could set up customized filters to narrow in on the donors engineered cells that are the most promising. In this particular case, I actually opted to do an additional analysis step where I plotted some composite scores for all of these different metrics in one graph. From this graph, we can pull out that this CAR T1B cells might actually be the most promising. And from directly within my dashboard here, I can see that the CAR T1B cell has this exact CAR gene. Now, if I click into this registered CAR gene, this will take me back to the registration page for this particular CAR gene. And now, if I click into this results tab, I will be able to see all of the data aggregated across all sectors of the organization for this exact CAR gene, from early reporter cell assays through CAR T production, all the way to donor cell testing. And in addition to showing the data themselves, if I scroll to the right in any one of these results tables, you'll see that it's also telling me who captured the results, what time they captured the results, and it is also giving me a direct link into the entry in which these results were captured. Thus, as a downstream translational scientist, I can jump into this CAR gene, see results captured by the most upstream lead generation team, and if I have any questions about how those results were generated, I can jump right back into that upstream entry and see all of the experimental details for how those results were captured. And that concludes my demonstration for today. So thank you for letting me share with you guys how Benchling can streamline and improve your CAR T R&D pipeline.